Hi guys, welcome. My name's Caroline. I realized I forgot to put that on my slide right now, so that's fun. But welcome to tonight's session on cellular energy. Thank you so much for joining. First, I hope you're all staying healthy and keep taking care of yourself, not just physically, but mentally and emotionally, because these are stressful times and it's really important to take care of your mental health. Hopefully you're enjoying maybe some good weather and you can get outside for a walk or something like that. But I totally understand if things are a little stressful or hectic. Hi, Sriveda. Thanks for joining again. I know some of you were with me earlier today. I did a Q&A session. So if you're coming back and just spending your whole day with me, well, thank you very much. For those of you who didn't join us earlier, I'm sure you've probably heard or listened to or seen or read or heard from other stressed out peers that the AP exam is virtual. It is 45 minutes long. It is free response only, and it may or may not, don't quote me, we're not positive yet, be open book. So those are the updates that we have. So very short, right? Used to be 3 hours. We're working with 45 minutes. We're working with all writing. So everything that we go over tonight, it's important to remember that you will have resources that can help you to use this information. You're going to have to write about it and make sense of it. Okay. So chances are we're going to be dealing with some kind of experimental design question. We don't know how many free response questions there are, but we'll get a lot of answers a week from Friday on the 3rd of April. As far as what that will look like. So stay tuned, but this is definitely a really important concept. So I'm going to go through basically a lot of the overarching concepts in Unit 3. That's going to be our focus tonight. All right, as always, follow us on all social media. I was telling my group earlier, we have a TikTok now too. I don't know how to work that. I don't do that, but if you do, follow us. Okay, so in this stream in particular, we're going to be going over the basics of cellular energy, just broad overview. I'm going to run through cellular respiration and photosynthesis, and then we'll look at a free response question that involves cellular respiration, and I can show you that. Before I dive in, does anyone have any questions about the exam or anything that's weighing on them right now that I can maybe help with? No? We're all feeling good out there? Okay, well, if anything comes to mind as we're going through, I can try my best to answer. I certainly do not have all of the answers, but can try to find them for you. All right, so at its most basic level, we know all things require energy, right? There's an entire unit on cellular energy. We all require it. And energy, we know also, is neither created nor destroyed. It's only transferred. So all the energy in our ecosystem, in our universe, in our biosphere, if you will, the entire Earth is not created nor destroyed. It's just transferred. So we get, obviously, our energy starting with the sun. And then that's going to be converted into usable energy by plants through photosynthesis. And then they turn that into glucose and other such products, which get broken down by plants and animals in cellular respiration, as well as all other living things. And that's going to be converted down into ATP, which is our cellular energy. Okay, so again, hi Derek, welcome back. This should all be reviewed to you, but just good to see these kinds of things and remember that energy is flowing through the ecosystem in this regard. And again, we're never creating new energy. It's just being transferred from another form. In order for cellular energy to work in an ecosystem, organisms need to rely on other organisms. We eat other organisms, right? So we're relying on them. And then reactions rely on other reactions, like cellular respiration relies on photosynthesis, and photosynthesis in turn relies on cellular respiration. And you can kind of see that here. The oxygen from photosynthesis is used in the Krebs cycle and in the final step of cellular respiration, the electron transport chain, and then that produces CO2 and H2O, which can be used as the reactants necessary for photosynthesis. So it is kind of a cyclical transfer of products and reactants from one reaction to the other. Olivia, great question. What's pyruvate? I will answer that for you when we dive into cellular respiration further. You may have heard it as acetyl-CoA, but same difference, and we'll take a peek at that. Soleil says, if we're reviewing, should we just follow the topics that you guys are posting about? Soleil, that's a great question. So I'm going to be posting I'm not sure if it's just for crammers or not. It may just be for crammers. Oh, that's a great question. I'll get the answer for you. Guides on how you can be studying. But the main topics you should focus on are units 1 through 6. And I would just go back and look at all of our old videos in order to identify those topics and the things that you might need to work on. We'll also be releasing study guides shortly, hopefully this week, for all of those units. So I would definitely use like the 1 through 6 study guides in order to figure out what content you know and what you don't know. Remember, all of that is just a jumping off point that you can then use to further your knowledge and find other videos, whether it's through Fiveable or through some other site, in order to get the support that you need. But everything that I think study guides are for everyone, Derek. I'm pretty positive we're going to make the study guides free for everyone, which is pretty awesome. They're really great and they work through all of the content. And then you guys can get, again, obviously in a 10-page document, you can't write everything that is that unit of AP Bio, right? But it's a good jumping off point for sure. So I highly recommend those. Tim, that's a great question. How much detail should we know about the content? You need to understand the content to the point where you know why things are happening. So I know we talked earlier about the possibility that this might be an open book exam. Again, don't quote me on that because we don't have a finite answer, especially for bio, of whether or not that's true. But regardless of whether it is or not, you need to understand things enough to be able to say, I understand why an electron carrier drops off an electron in the electron transport chain and what that does in order to create ATP. You can look at a textbook and you can see that that electron is going to help to contribute to make ATP, but you need to understand why. So as you're going through things, you should always be asking yourself why things are happening and make sure that you can explain it in an easy enough way that someone else could understand you. So I don't have a, so Derek, you don't need to know any of the exact values. Even if this test was the regular 3-hour test, you wouldn't need to know the exact values. And I'll show you an example of why that is true. But you do not need to remember that. You just need to understand why things are happening. So as we go through, I'll tell you what you should and what you don't need to know. And hopefully that puts some minds at ease, because it's really just the big picture topics that you need to understand. And if you have any questions throughout, obviously I can answer them for you. Okay. So as we already said, cellular energy is ATP. So this is something that you should be familiar with, right? I always think it's really important to understand why our cellular energy is ATP, and it all resides in this third bond here. So we have this, it's adenosine triphosphate. So here's the adenosine group. And then we have 1, 2, 3, let me put my little pointer on. 1, 2, 3 phosphate groups, okay? And this third bond here, the one that's connecting this last phosphate group, is super high energy. Bonds carry energy in them. And then when that bond is broken, which we can see here, we have a negative delta G, which means we're releasing energy. We can do cellular work, okay? So that energy that's released from that phosphate group actually allows us to do work in the cell. Okay, because remember, energy is not destroyed, it's actually going somewhere else and it's going to stimulate another reaction. ADP, adenosine diphosphate down here, has just those 2 phosphate groups and is easily recycled by adding that phosphate group. Okay, this is the condensation reaction. We're condensing, we're going to get rid of a water molecule. And this happens multiple times through glycolysis, the TCA cycle, which is the same thing as the Krebs cycle. Oxidative phosphorylation and photosynthesis, any reaction that creates ATP is doing so not out of thin air, but it's really just phosphorylating or adding a phosphate group to ADP. So these things get recycled. And this is also why reactions rely on one another, because once that ADP is used up, it needs to go back and be recycled so that it can be made back into ATP. Otherwise, we run out of our ingredients, right? And that would be no good. So hopefully that makes some sense, because understanding the basis of ATP will help you a lot in understanding these reactions and just how energy in our cells work. Okay. Cool. So this is why I start. So I'm going to start cellular respiration and photosynthesis with what the purpose is, right? Because that's the big picture stuff that you need to start remembering, as opposed to the teeny tiny details, because those are not going to be super, super essential that you have them all memorized. And yes, Sriveda, you're exactly correct. Adenosine di and adenosine tri. So di means 2, tri, like a tricycle, means 3. So there's 3 phosphates there. Okay, so the overall purpose is to convert food sources into usable energy, right? And by usable energy, I mean that cellular energy or that ATP. So we're harvesting stored energy. So we can store, like especially plants, they'll store energy as starch. Like potatoes are all really starchy. That's full of sugar and carbohydrates. And that's a way of that plant, the potato plant, storing their energy for later use. And then we break it down. And then in most examples, we see that as glucose to ATP. Doesn't have to be glucose, right? But more often than not, when we see this, that's what we're going to represent it by. And then you can see our chemical formula here, glucose and oxygen yields carbon dioxide, water, and ATP. And it is truly the reason why we breathe. Sometimes I'll ask students like, hey, why do you breathe? And they're like, to get oxygen. I'm like, but why? They're like, because we need it. I'm like, but why? They don't know why. The reason is for cellular respiration. Why do we exhale carbon dioxide? Because of cellular respiration, right? So it's all related. But those two reactant and product pair are obviously super, super important. So that's our overall goal here. And it's really important to not lose sight of the fact that plants also use cellular respiration, right? They obviously go through photosynthesis, but that just makes these food sources, this glucose or starch or whatever it may be, they still need to break it down into ATP just like we do. So they're capable of doing both of these processes, which I think sometimes we lose sight of. Okay, so there's 3 major steps, and this is one of those things where I'm going to say you do not need to memorize every little part of all of these 3 major steps, but you do need to be familiar with them and why they're important. And again, I will show you a free response that hopefully makes more sense of this. So we have glycolysis and pyruvate oxidation. Okay, so that's where we'll talk about what pyruvate is. Then we have the Krebs or citric acid cycle. Most people call it the Krebs cycle. Some call it citric acid, same difference. And then lastly, the electron transport chain, which is where we're going to make the majority of our ATP. So some really important things to remember here is that glycolysis is the most evolutionarily ancient pathway, and that should make sense. It comes first. So it was the first thing that evolved. That's what I mean by evolutionarily ancient. And obviously, evolution is not going to be on your AP exam because it's in Unit 7, but it's still important to remember because I still could pull something like this is Unit 3 right now, and we're talking about evolution. But this evolved first, and we can show that it's done by all living organisms, everything. It can occur without a mitochondria. It can occur without oxygen. So even the bacteria that are living in the inside of your gut where they actually have no access to oxygen. Oh no, it did? I wonder why my computer's doing that. Guys, I'm so sorry. Be prepared. I might all of a sudden sound like a demon. I'm not sure why. Because I wasn't too close to the camera that time, right? I wonder if it's when I switch back and forth. I don't know. I do apologize, though. I'm not sure why that's happening. But it's done by all organisms, okay? So also don't lose sight of this. Like our little bacteria friends are also doing glycolysis. And if they asked you something about like an ancient pathway or something along those lines, you're going to be talking about glycolysis. And that hopefully makes sense because it's the first anyway. So it would have evolved first. Okay, so here's what it looks like. And the reason I'm showing you these pictures, so when you see these black and white images, these are actually snipped from a College Board free response from 2015. So they gave students all of this information. So that's why I'm telling you, you don't need to memorize it, right? You don't need to memorize these numbers, that it's 2 electron carriers and 2 ATP and whatever. You really just need to remember the process and what's occurring here. Okay, so what is occurring here? Well, we're breaking bonds, and that's going to do 2 things for us. So when we break a bond, we're going to release energy, because remember, energy is stored in bonds. We just talked about that with ATP. And we're also going to release electrons. So these are the kinds of things that you need to be familiar with, not memorizing numbers. Okay, so if they were to ask you where the energy comes from in glycolysis, you can say the breaking of the bond releases energy, and it also releases electrons from those that were shared in the molecule, and those are picked up by electron carriers. And if you're not familiar with what an electron carrier is, literally carries electrons, but our example of that is usually NAD+. And then in bio, we're going to carry that with a hydrogen ion. So it's like a proton, H+, and an electron pair up together. And that's how we're carrying this guy. So you can see that they are picking up electrons as the bond of glucose is broken into this 3-carbon molecule. So that's what these little dots represent, carbons. So we'll start with 6 carbons in glucose, and then we go to 2 3-carbon molecules. So we still have 6 carbons. Matter cannot be created or destroyed. And we call that molecule pyruvate. So that's what that is. It's kind of the end process of this glycolysis situation. And then we go one step further and we actually oxidize pyruvate. So remember that oxidation is loss. Hopefully you know oil rig or Leo the lion goes grr or something like that. I like oil rig. So oxidation is loss of electrons. So we're losing more electrons here as we're breaking bonds, and we're releasing some carbon dioxide. So 2 carbon dioxides that we will exhale. We're making a few more electron carriers because we're releasing electrons from those bonds. And then we're ending up with a compound called acetyl-CoA, which has a much longer name that you don't need to be familiar with. All you need to know is that there's 2 of them. They have 2 carbons. The other 2 carbons were exhaled as CO2. That's a toxic waste product for us. And this will enter into the Krebs cycle. Okay. Can FADH2 be used as well? It can be used as well. It is used in the Krebs cycle, which you don't need to explicitly remember, but it is another type of electron carrier. And then when we talk about photosynthesis, we'll talk about NADPH. Does pyruvate oxidation happen in the cytoplasm or the mitochondrial membrane? It will happen in the cytoplasm, Derek. That's a really good question. Oh, what did oil rig stand for? Sorry, Reba, let me type that out for you. If you're not familiar, maybe you haven't had a chemistry class, or maybe your chemistry teacher used a different acronym. Oil is for oxidation is loss. Okay, and then rig is for reduction is gain. So if a compound is gaining electrons, we say it is being reduced. And if a compound is losing electrons, we say it is being oxidized. So if they tell you that something is an oxidation, right, then you know that oxidation is loss, and then that thing is losing electrons, usually because bonds are being broken. Hopefully that helps, Reba. Okay, any other questions about what's going on here? Hopefully this is good review. And again, the more important thing is to shift your brain into thinking about where the energy is coming from, where the electrons are coming from, and why that's important, because in this free response, all this information was given to you. You just needed to expand upon it. Okay, and don't worry, we'll practice a free response because that's what your exam's gonna be. I have hair stuck in my eye. Okay, so just in summary, what goes in? Glucose, what comes out? ATP, electron carriers, and then pyruvate, which is oxidized into acetyl-CoA. And you guys should have access to these slides, so it kind of simplifies things, which is kind of nice. And you can see the ATP, and then I think of the electron carriers as little piggy banks because they're storing electrons, they're storing energy. It doesn't mean anything for us right now, but once it gets to the electron transport chain, which we'll talk about in a little bit, we're going to get a lot of bang for our buck from those electrons. So they're kind of just going into storage for the time being. Okay. Any other questions on glycolysis and pyruvate oxidation before we move on to Krebs? Actually, I think we're moving on to fermentation. So for the most part, Soleil, an electron carrier just holds 1. So we have NAD plus going to NADH, which would be 1 electron. FADH2 can carry 2, but otherwise they each carry 1. That's a good question. Any other questions? Okay. But back then. Okay. So this was the next part that I wanted to talk to you about, and this is the process of fermentation, which you guys have probably heard about. NADH and NADPH striveta is just the organism that uses it. So NADH is used in cellular respiration and NADPH is used in photosynthesis. I just remember P for photosynthesis or P for plant. I don't know what it actually stands for. Something chemical probably, but they do the same thing. So Not too hard to remember. Okay, so the big takeaway that I want you to get here is we think back to our piggy bank analogy. If we fill up all of our piggy banks, then we can't collect any more money, right? We don't have any way to collect more money. And so all the money that we're making is going to waste, right? Because our piggy banks are all full. So we need to empty those piggy banks in order to refill them with more money, right? Hopefully that analogy makes sense. I try to come up with weird ways to remember things because that's what sticks in our brain. So for us as aerobic oxygen using organisms, we naturally recycle our electron carriers when they drop off their electrons or their money at the electron transport chain. But things that are anaerobic, that live where there is not oxygen, are unable to do that. So they have to drop their electrons off on other products in order to empty their piggy banks or their electron carriers in order to refill them. So that's where we see down here, this is the process of a couple different things dropping off electrons on different products to make things like ethanol, which is alcohol. So fermentation is how beer and wine are made. So yeast or other bacteria are used and they're put in an anaerobic, which means without oxygen environment, and they're given sugar and they break it down and they need to recycle their electron carriers. So they produce this byproduct. Also lactate or lactic acid, we can do that if we run out of oxygen. So if you're working really, really, really hard, maybe you're sprinting and your muscles run out of oxygen because they've used it all up, they can start going through fermentation and producing lactic acid, which makes you sore. So just know that species who either don't have enough oxygen or aren't exposed to oxygen at all can still recycle their electron carriers, which again, these are the big idea pictures that you should be familiarizing yourself with, but they just produce these byproducts in the process because they're dropping their electrons off on something else. Tim, what are the locations for everything? So glycolysis and pyruvate oxidation occur in the cytoplasm of the cell, and then our other two stages are going to happen in the mitochondria. Bacteria can still do them in a modified way, but it's not going to be as successful as ours is. They can kind of have like a little modified electron transport chain, but it's not like ours is in the mitochondria. Do you have to know where in the mitochondria? So I'll show you where. There's like the mitochondrial matrix and then the mitochondrial membranes. But it should make sense once we talk about like the electron transport chain has to happen on the membrane versus the Krebs cycle, which can just happen in the inner space. Yeah, exactly. But again, I wouldn't expect you to have to know that much detail. But you know, it's a new year, so. Here we go. Okay, so then going off of Tim's question, then now we've transitioned into just the matrix, which would be like the gooey in between. So if this is my mitochondria and I have all these little membranes, the in between my fingers would be where the Krebs cycle is taking place. And the Krebs cycle involves so many things and so many enzymes. And if one day you decide to become a doctor, you will memorize all of those enzymes. But for right now, you don't need to. All you need to know is the general overview of what's happening here. So basically, we're bringing in that acetyl-CoA, and remember that these two carbons, it's actually 4 carbons, right? Because we have 2 of them, came when we started to break down glucose. We made pyruvate, and then we made some CO2 and some acetyl-CoA, and they're going to get just transformed by some enzymes, and they're going to go through this cycle of transformation of making and breaking bonds. And those are the details that you do not need to know. What you do need to know is that in this process, as we're making and breaking bonds, we're going to be releasing carbon and we're going to be releasing electrons and a little bit of energy. So what you can see here is we have 4 carbon dioxides being released. That's our remaining 4 carbons, right? We had 2 carbon molecules left. So all 6 carbons that we started with have now become CO2. So they've actually tested this before, and they've given people radioactive glucose to eat. Not harmful, but they actually eat the glucose, and the carbon in the glucose is radioactively labeled. And then those people will, eat it, digest it, and then they'll measure the radioactivity of their breath, and they can detect all of that radioactive carbon coming right back out of them because the carbon just gets broken down, converted into CO2, and gets released. Obviously, we have a lot of other carbon sources in our body, but specifically the ones in glucose will get converted into carbon dioxide. So also in this process of continually making and breaking bonds, we're releasing a lot of electrons, and that's the number one thing that you need to remember about the Krebs cycle. We are releasing a massive amount of electrons. You can see we have 8 electron carriers being made here, all being filled up with electrons that have been released from this process. And that's why the Krebs cycle is so important, because those electron carriers are going to make us lots and lots of ATP in our final step of the electron transport chain. And you can see that we also release a tiny bit of energy here, 2 GTP, which is basically the same as ATP. And I should have mentioned that with glycolysis too. That's a very small amount of energy. So with this Krebs cycle, what we care about way more than just those 2 GTP is the immense amount of electron carriers that will then go make more ATP in the electron transport chain. Any questions on Krebs? Krebs is my favorite because it's super straightforward. Okay, so just as a reminder, your summary, we're putting pyruvate in and we're getting out a little bit of, I said ATP, GTP, whatever you want to call it, lots of electron carriers, and then CO2, which again is just a toxic byproduct that we are releasing. Okay. Then lastly is where we really start to make all our money, and it is the electron transport chain and chemiosmosis. So here, again, just knowing your chemistry, knowing a little bit about cell transport, that's all going to help you here, and knowing about concentration gradients is really, really important here. So those are things to remind yourself of before you take the test. So electron transport chain, this is where we're going to drop off those electrons. We're emptying the piggy banks. Okay, so we drop off the electron, and the electron is going to move through the electron transport chain, which has these 4 complexes, which I think of kind of as like a conveyor belt. They basically get more and more electronegative as you go. And so they're dragging the electrons through, which is giving us some energy, and it's giving us the energy to pump hydrogens across the membrane. Okay. So you'll notice there's more hydrogens out here. This is where we have a high concentration of hydrogens, less hydrogen in the inside. So we're going against the concentration gradient right now from low to high. So that takes a little bit of energy, but we get that energy from the electron that was dropped off. Okay, so that's helping us to create this concentration gradient, which is very, very important. We'll look at that in a second. As that's happening, as this electron's getting pulled through, it will finally get dragged to the most electronegative of electronegative atoms, oxygen, second most electronegative, but the most electronegative thing in this diagram, and it will bind with oxygen. Remember that electrons form bonds, so oxygen is going to meet up with some of these protons that are floating around to make some water. If this guy is not here, so if you stop breathing, right, this entire chain stops. It's like there's a tree in the road, right? And we are unable to continue, which means we are unable to pump hydrogens, which means we are unable to make ATP, which means you are unable to survive. Okay, so the fact that oxygen is here waiting because you breathed in is a very important thing, because it keeps this entire process going. And that is why we breathe. So you can tell someone that tonight as a fun fact. Okay, so that's what's going on over here. So electrons are being pulled through, energy is pumping the hydrogens across their membrane, or across their concentration gradient, my apologies, and then it's ending up on oxygen and forming water. Do we need to know how many H plus need to pass for the electron to move past each complex? Oh, definitely do not need to know that, Sriveda. Way too specific. Soleil, what does electronegative mean? That's a great question. So electronegative is a chemical structure's desire for electrons. How much do they love electrons? How much energy does it take to remove an electron from them? And so things like oxygen really love their electrons. They're very electronegative. It really depends on the group that you're in the periodic table. So things that have like 7 electrons, they really just want 1 more in order to fill up their octet. And so those types of things like oxygen are very, very electronegative because they're looking for more electrons in their life. Hopefully that's my biological answer to your chemical question. Okay, so we haven't even made any ATP yet. Any questions about the electron transport chain though? Of course, that's what I'm here for. Thanks. You guys have been so great. Everyone who was here earlier too at asking questions, I'm very thankful because it's very boring to just talk to myself. So keep them coming. That's, this is your learning opportunity for sure. Okay. So then on your right here, you see this lovely little pump called ATP synthase, which I'm realizing, oh no, it is labeled. And we know anything that ends in ACE is a what? Someone write it out. Anything that ends in ACE, what is it? An enzyme. Yes, you guys are rock stars. Perfect. Remember that. It's a great way to remember. So anything that ends in ACE is an enzyme. This is an enzyme, ACE, that synthesizes or makes ATP, right? So you can never forget what these things do because they're named right according to what they do. So basically, and you don't need to know all of the details of this, but basically you can think of this as like a water wheel or a windmill, and it's actually going to use the kinetic energy from the flow of hydrogens in order to create ATP. So let's back up a minute. Remember that we have a high concentration of hydrogen ions on the outside of this matrix right now. Obviously this is happening in the membrane now of the mitochondria, right? Because we're in the membrane. So we have a high concentration out here. These guys can't get through the membrane on their own because they're charged, right? Nothing charged or polar can get through the membrane on their own. It's a good combination of units here, going back to unit 2. Okay, so these guys have to get through some sort of protein channel, and the protein channel that's going to allow them to come through is ATP synthase. So they are super pumped to move and spread the heck out and go back down into a low concentration. Okay, and this is actually going to like spin, you can watch videos of it, of what scientists have been able to model, but it's going to kind of spin a crank almost, which is going to create kinetic energy, which can be used to phosphorylate. Remember, phosphorylate just means add a phosphate. So we're phosphorylating ADP, adenosine diphosphate, and we're making ATP, adenosine triphosphate, and some water. Okay, so this is the big money maker. And as the hydrogens come rushing through, we're cranking out a ton of ATP, depending on who you ask, anywhere between like 32 and 36 ATP for each glucose from this process. So in comparison to the 2 that we make in glycolysis and in Krebs, we make a ton more through this process with ATP synthase at the end, and this process of chemiosmosis. So this is another important vocab word to remember. Obviously you can have notes, but you want to make sure that, or we think you can have notes, but you want to make sure that you have them handy. And osmosis, remember, is just the movement of water. So I just think of this as the movement of like chemicals or ions, chemiosmosis. But the process of moving things from a high concentration to a low concentration is what's happening here, chemiosmosis. Any questions about that? If you guys feel confident about everything that I just said, then I feel very confident in you, because this is not easy stuff. I think photosynthesis is way easier. So this is good stuff if you guys feel solid. Any questions? Sugar, when referring to sugar, what exactly? Oh, glucose? Yeah. Glucose is sugar. Anything that ends in ose is a sugar. So sucrose is like table sugar, like what you would add in your coffee or cake. But yeah, hopefully that answers that question, Angela. Thanks for joining again. Okay, no other questions? All right. So just your final summary then, electron carriers and oxygen go in, so they're going to drop off their electrons, and this is why we're breathing, and then ATP and water come out. Lots of ATP. And water, remember, water is made when oxygen and the electron and the hydrogen atoms all come together. Okay. So let's jump into photosynthesis then. This will be quick and easy, and then we can practice a free response, and hopefully you guys feel confident by the time we get done here in about 25 minutes. Okay, so the purpose of photosynthesis is to convert light energy into chemical or usable energy. Light to sugar is the general process here, light to glucose, okay, or light to energy, because it's not always glucose, but we like to oversimplify things in science. Someone once told me that as you progress in science, you just get lied to less and less. And I think it's true, because you just like find out that what you've been learning is not nearly as accurate as what actually happens. Okay, so the big thing here is that basically all energy on Earth comes from plants. We know this from producers, right? So that's getting into ecology, which you don't need to know about. But that's where all of our energy is coming from. Even if you never eat a plant, the thing that you ate a plant, and that's where that energy came from, right? Because again, we're not creating or destroying energy, we're just transferring it, and it's all originating from the sun, which I think is kind of fun to think about. So our generalized formula is the exact opposite of cellular respiration, but they are not just like a reverse process. It's not just cellular respiration run in reverse. They're different processes, but it is lovely that they have the exact reverse formula. So we're putting in CO2 and water, and we're getting out glucose and oxygen. Japesh, this is somewhat off topic, but a reduction is when a compound. Yes. So that's that oil rig, or maybe you learned Leo goes grr. Loss of electrons is oxidation, gain of electrons is reduction. I like oil rig. Anyway. Okay, so there's your formula. And this one only has two major steps, the light reactions, or sometimes they're called the light-dependent reactions, and then the Calvin cycle, sometimes referred to as the light-independent reactions because they can happen without light. Those are the two that we're going to take a peek at. Oh, look at that lovely diagram. So colorful and beautiful. Okay, so to preface this, because I think Tim asked earlier, and I realized I didn't really preface this very well for cellular respiration, for photosynthesis, this is all happening within the chloroplast of the plant. Or we have photosynthetic bacteria, some photosynthetic bacteria called cyanobacteria. But we do not have a chloroplast, so this does not exist in us. But the chloroplast is just like a little oval, what's the word I'm looking for? Does anyone else feel like their brain is working maybe half speed with being in quarantine? I feel that way. Anyway, we have some sort of oval structure called the chloroplast, and then within that, we have little stacks of membranes that I think look like pancakes. That's how I like to remember them. And those are called the thylakoids. And then we have just the stroma, or the space, which I like to think of as the syrup in between. Okay, and I should have put a picture up there for you. Okay, so that's our lovely chloroplast. And what we have here is obviously another membrane, and it's a membrane that's this is essential to. So it's a thylakoid membrane, so one of the membranes of those pancakes that is essential to the light reactions. Okay, there's a lot of moving parts to keep track of. Again, I just want to reinforce, you do not have to have all of this memorized, but you should understand why things are happening. But the things that are happening in photosynthesis are the same things that occurred in cellular respiration. Right? We're making ATP, we're making electron carriers. We know all about this at this point. You guys are pros. So, general overview is that we have light, and light is both a wave and a particle, if you take like electricity and magnetism one day for physics. But these particles and waves of light are coming down all the way from the sun, and they are striking these little proteins that are embedded in this thylakoid membrane, okay? And they're called photosystems. So they're labeled PS2 and PS1 for photosystem 2 and photosystem 1. Why does PS2 come first? It was discovered second, right? So they just named it second, and just instead of, renaming it to save AP Bio students for years to come, they just said, we'll just call it photosystem 2 and make it go first, just to sprinkle a little bit of confusion on top. Regardless, here they are, they're sitting here, and they both basically are constantly, when the sun is out or when, even when it's cloudy, there's still beams of light coming through, get struck with these photons of light. And what that does is it makes an electron really excited. So if you've taken chemistry before or any class like that, you know that electrons actually can get super excited and they move up into a higher energy level, right? It's kind of like you see a celebrity, they like freak out and jump up into a higher energy level. Okay, so that's what's happening when light is striking these photosystems. We're actually basically like popping off an electron, right? Because it gets super excited and it hops off into a higher energy level, and the photosystem actually loses that electron. Okay, so as these electrons get lost, they're going to do two different things. So this one's super easy, so we can talk about it first. So the electron that's lost from photosystem 1 just gets picked up by an electron carrier. And we were kind of mentioning this earlier, but the electron carrier that plants use is NADP. And again, I just remember P for plant or P for photosynthesis, but we're going to pick it up and then it gets filled up just like our piggy banks from cellular respiration, and we're going to use it later. Okay, so that's what happens to this electron. Electron carrier made, good to go. We know all about that now. Hopefully that's familiar in your brain. And then this electron is actually going to go through a little, and you don't need to know all of these things, way too many words, but it's going to go through its own little electron transport chain. And what you can see happening here is the same thing that happened in the electron transport chain in cellular respiration. We're pumping hydrogens across the membrane. But in this case, we're still pumping them from low to high. We're just pumping them inside of this thylakoid lumen, okay? So you can see the hydrogen, it's coming in against its concentration gradient because of the electron transport chain. I'm gonna pause there for a second. Any questions? Yes, that is the name of the pigment. Thank you. So they have slightly different pigments on them. So you can see there's slightly different shades of green. And that's because they absorb a slightly different wavelength. It's going to be the wavelength. They're either absorbing 680 nanometers or 700 nanometers of light. Yeah, cool. Any other questions about what I just said? So we understand why the electron carrier gets made. We understand why the hydrogen gets pumped. We feel okay? Someone has a question. Oh, Reba, thank you so much for asking. So Reba says, I'm confused how NADP gets to NADPH. That is a great question. So NADP is an electron carrier. So think about like an empty piggy bank, right? And it is just waiting to get reduced. Reduction is gain. It's waiting to grab onto an electron. So an electron plus a little hydrogen ion come together and get picked up by this piggy bank. So it's just an electron carrier. That's its job, right? It's just really able to grab onto and carry these electrons to where they need to be, just like NAD and NADH did for us in cellular respiration, where they pick them up and then they bring them to the electron transport chain. That's what happened in cellular respiration. Here, they're going to pick them up and bring them over to the light reaction, but they just carry the electron. And in bio, that electron doesn't just get globbed on. It actually gets globbed on with a hydrogen ion to make like a whole hydrogen atom. So that's why we have the H as opposed to just like NADP minus or something like that. Hopefully that answers your question, Reba. Okay. Why does the hydrogen get pumped to form a concentration gradient so that it can power? Where is it getting the energy to bring? Oh, the electron. Derek, thank you. You're like a TA in the room. That's exactly right. Everything Derek said. So same with the electron transport chain before. As these electrons are moving through the electron transport chain, it's giving us enough energy to pump the hydrogens against their concentration gradient. And then we're going to utilize that on the right-hand side to make ATP. Okay. All right, thanks guys. You guys are great. So hopefully that part makes sense. So then just two more parts. We're going to talk about ATP synthase. But before we do, this photosystem has been robbed, right? This electron got so excited, it hopped off, it ran away from home, and PS2 is very sad, right? So we got to replace that electron, okay? Because otherwise we have this kind of unstable protein and we don't want that. So how do we do that? We're actually going to break apart, and by we, I mean the plants that do photosynthesis are going to break apart a water molecule into hydrogen ions, which you can see are going to be used just like all the other hydrogen ions, and oxygen. Okay. And remember, when bonds break, electrons are released. So the electron that's released will replace the one that was lost, because we want no lost electrons in the end. Okay. PS1 also lost an electron, as you guys remember, to make this electron carrier. But this electron that popped off of PS2 will go ahead and replace the one that was lost on PS1. Okay. So PS1 loses an electron. It's replaced by the electron from PS2. The electron lost from PS2 is replaced by an electron from water. And then we get oxygen. And, plants think that oxygen is a toxic byproduct, so they're just going to release it, similar to how we just release CO2. And we're very thankful for them because we can breathe another day. Okay, last thing that happens is all of these hydrogen ions are going to go through ATP synthase in order to make ATP. And we know this at this point, right? So ADP and phosphorus will bond together to make ATP using the energy from the hydrogen ions flowing back with their concentration gradient. And everything looks upside down from what we just saw in cellular respiration. So you can just tilt your head upside down and you'll notice that really the same exact thing is occurring. The electron transfer chain does the same thing, and ATP synthase does the same thing too. So is this ATP synthase and photosynthesis and cellular respiration the same? Yes, Angela, it's exactly the same, which is great. Same enzyme does the same exact thing, uses the hydrogen concentration gradient both times. Tim, what's the interrelationship between photosynthesis and plants' respiration? So photosynthesis is going to make the sugar that plants will then just break down, but they're basically providing themselves the products and reactants necessary. I think I don't have a picture. Oh, I might have a picture at the end kind of showing those interrelations. So they're going to utilize, the glucose and oxygen from this in order to break down the glucose into ATP. Okay, so what goes in? Light, water, and then I wrote this out, and I should have written it for, all the others too. We need these empty ADP and NADP plus, right? We need our empty piggy banks, and we need our empty ATPs to be refilled, right? Otherwise, it's like what I said, we run out of piggy banks. We filled them all up, and we stop. We have to throw away the rest of the money that we make, right? So we need to empty those guys, which is what we're going to do next. And then what comes out? We know we have some ATP, which we're going to be using, some electron carriers, which we're going to be using, and then oxygen, which again, we're very thankful for. Okay. Any more questions? Yes. So Soleil, the ATP and electron carriers are going to go out of the thylakoid and into the the stroma, just like the inner membrane space. It's still inside of the chloroplast, right? But it's just here. Let me pull up a picture really quick of a chloroplast, which I should have had for you guys. My apologies. I didn't have a picture of a mitochondria either. I'm losing my mind. All right. So let me share this screen really quick just so we can take a little peek. Woo. Hopefully I don't do the devil voice again. Seems like it happens when I'm switching screens. Okay, so here we have the inner membrane, the thylakoid, the stroma, the inner membrane space. So the stroma is like the space in between these little pancakes. Okay, so the thylakoid is where the light reactions are happening, and then this space in between is where our light independent reactions will happen, also known as the Calvin cycle. Okay. Okay. Cool. Let's bat back. So still all happening in the chloroplast. Okay. All right, so next is the Calvin cycle. And we may end with this, but I'll leave you with some free response homework that I can look at for you. I'll post it in the community group, and then you guys can write your responses for me, and I'll grade them for you. So Calvin cycle is what's next, and we're going to utilize the ATP and the electron carriers that we just made, and we're going to make that sugar. Okay, so here's what happens. Again, this is one of those other things very similar to the Krebs cycle. You do not need to know a lot of this unless you want to become, I don't know, a ecologist one day. Okay, here's what you really need to know. Carbon dioxide, that toxic gas that we breathe out, plants use it, and they use this special enzyme called rubisco that turns this inorganic substance into an organic carbon-containing compound that we can start to work with. So it's going to pull it out of its gas form, and it goes through this process of carbon fixation using the enzyme rubisco, which doesn't end in ase, but it actually does because it's ribulose bisphosphate carboxylase, which is a very long name, which is why we call it rubisco. Okay, but carbon dioxide and this enzyme rubisco are going to be combining together in order to make an organic or usable form of carbon. Okay, so that's step one. Then there are a lot of other steps that you don't need to be familiar with. Like you don't need to know the reduction phase or the ribulose regeneration or any of this stuff. Okay, all you need to know is that we are going to convert this carbon dioxide, which we've just kind of fixed into something more organic, into a product like glucose. Okay, so think about that. We're going to need to make a lot of bonds, and we're going to need to use energy in order to do that. We need electrons, right? If we're making a ton of bonds, we need energy and we need electrons. Okay, so where are we getting that energy and those electrons to take this single carbon molecule into a 6 carbon molecule? We just made them in the light reactions, right? So all that ATP and all of those electron carriers that we just made are going to come and be used right here in order to convert a very small inorganic carbon dioxide into a very large organic sugar molecule. Okay, so again, the energy and the electrons for that process come from the light dependent or light reactions. Okay, and they're given to this cycle in order to be used. Okay, what you'll see here is something called G3P, glyceraldehyde 3-phosphate, and we see it up here again, phosphoglycerate, kind of same difference, but glyceraldehyde 3-phosphate, this is then going to go be used in making things like glucose, starch, cellulose, all of those things that plants use. If you want to remember that it just makes glucose, that's fine too. This is just a little more specific, that we're actually making a compound that can become a number of other carbohydrate compounds like glucose, starch, cellulose. Those would be the big ones that plants use. Okay, but we're taking carbon dioxide, we're using this enzyme rubisco, and then we're using the energy and electrons from the light reactions in order to make glucose or something similar. Okay, the important thing about Calvin cycle is it also recycles our electron carriers and our ATP. So we get ADP and NADP plus, which then can go be refilled at the light reactions. So we're emptying our piggy banks, which is important. So then we can go refill them and the cycle can continue. Any questions about that? They're not the same thing, but the answers given there are correct. And you do not need to know all of the crazy acronyms here. Cool. Yeah, rubisco is an enzyme, so it's a protein. So the cycle does occur twice because we need 2 G3Ps in order to make a glucose. That's why. So hopefully that answers both of those questions as to why the 3 carbon G3P or the 3 carbon molecule, we need 2 of them in order to make a glucose. Okay. Any other questions? You guys are rocking and rolling. Okay. So since photosynthesis, are they using the exact same one? So it would be the same enzyme, but it's in different locations. So even for a plant who's going to be using ATP synthase in two different things, they're going to have one in the or many millions in the mitochondria and then millions in the chloroplast. So same enzyme but different location. So remember that. Because remember, plants also have the mitochondria, which is where their cellular respiration process is occurring. Okay. Derek. Oh, because we're constantly, so that's another really good question. This is what gets into the details of the Calvin cycle. We're going to break that bond even more and build it into, we're going to build it into other molecules throughout the course of this cycle. Kind of like how I said, we're making and breaking in the Krebs cycle. We have a lot of different enzymes that are going to make this into a couple different products, but the stable one that is able to be converted into other stable energy producing carbohydrates is G3P. But don't worry too much about all the chemistry behind it, as long as you're familiar with why these things are happening, what the energy and electrons are being used for to make these bonds, and then what our end product is. Okay? You guys are asking great questions. So just your reminder is that we're making glucose, NADP plus, and ADP. These are really important because they're going to go back to the light reactions, and we're putting in these compounds here. Okay, and then just kind of our overall review. So this is how energy is kind of moving through systems, right? We have sunlight, carbon dioxide, and water going into the chloroplast, and out comes glucose. And glucose is broken down in the mitochondria, well, not in the mitochondria yet, but we're going to go through the process of glycolysis, love the typo here, to make a little bit of ATP. Then we go through the Krebs cycle and the electron transport chain inside of the mitochondria with a little bit of oxygen, and we make ATP. We also make these products to go back up here, and the cycle continues. Okay, so if you're a plant, you're doing this constantly. We just do this part down here by eating glucose and things that have glucose in them or have eaten glucose. Okay. Okay, so I think I was showing the group this earlier, but I don't remember. So I'm going to post this for you guys. And you're going to be looking at the long free response question 2 on this. It's all about cellular respiration. I'm also going to post it as a community thread to Fiveable tomorrow. Okay. Just because that's when I'll be going through this unit. And when I do that, I will send you guys an email to everyone who attended tonight saying that the post is live, and you can actually answer the post on the community page, and I'll see your response and I can give you feedback on it and tell you how many points you would have gotten. Obviously the rubric exists out there, right? So, don't look at the rubric. Just try to do this on your own and see what you can get. And then you can write that on the board tomorrow, and I'll give you guys some feedback on it if that's something that you think would be helpful. It's a pretty good application of this cellular energy information. And remember, your AP exam is going to be all free response. So writing is the most important thing. Okay, so you can get a head start on that if you want. I'll have that live and ready for you tomorrow, and I will make sure to send you guys all the link to it, okay, to make sure that you guys get it and can participate if you would like. Will there be a game of fives for AP Bio soon? Hopefully there will be. It's a great question. Derek, my email, I'll post it here in case you guys have questions. You can shoot me an email, but I'll be sending you an email just like through Crowdcast so that I can make sure that you guys all have it, because you guys sign up with Fiveable with an email. And Sriveda, I hope so. I think one's in the works. And if I get details, I can also send that out to the group. Thanks for asking. So as a recap, I don't know what next Wednesday's topic is going to be yet. So stay tuned. We're going to finalize that tomorrow. But I do know that on Monday evening at 8 p.m. Eastern, I'll be running my first cram session, which will be a Unit 1 review. So Allison, it's Wednesday at 8 Eastern. So if you're in Central Time, it will be Wednesday at 7 will be the next free session. But my cram session is on Monday at 8 Eastern or 7 Central. And that's going to be all of Unit 1, but that you do need the cram pass for. We'll go into lots of details with Unit 1 and what you need to know. Okay, so stay tuned. Hopefully I see some of you there. If not, I'll be streaming, one of these Wednesday nights coming up, or maybe doing a Q&A session again soon. Have a great night. Take care of yourselves. Take care of your family. Get a brain break, especially if you guys have been all over like College Board's posts and all of these posts all day. So make sure that you take some time for yourself too and your family. And let me know if you have any questions, and I'll shoot you guys that email tomorrow. Bye, y'all.